All right, everybody, welcome to week 2 of the semester. Let me share my screen with you real quick. And what we will do is jump right into biology of mind and behavior. So this is what your brain looks like. Today, we're going to talk about neurons, neurotransmitters, how your brain works, the different lobes, all that really exciting stuff going on in between your ears right now. So your brain is composed of 100 billion of these. What are they? They're neurons. They're the cells within your brain, and they spread throughout your body as well. They are bundled throughout your spinal column that relays messages throughout your body. They extend all the way to your fingertips, so you can feel things, touch things, sense things, temperature, like all of the wonderful experiences that that we have. It's basically all made sense through neurons relaying the sensations from our outside world through our fingertips, through our spinal cord, up to our brain, where we process and make sense of those messages. And sometimes the response is, hey, this stove is really hot. I need to pull my hand back. So those neurons are also involved in that process of relaying that message from your brain to your spinal cord, to your hand, to move those muscles, those motor neurons away. So anyway, we're going to talk all about neurons and the brain today. So the first off, here's a really cool picture that kind of shows what these neurons are looking like when they're firing. Firing is just a fancy word for saying that those neurons are relaying that message. So for your notes, what is a neuron? It is a cell. It is the basic unit of the nervous system. So there are a few different types of neurons that we'll talk about. They are involved in like receiving signals from neurons or sensory organs, like do you feel full? Do you feel hot? Do you feel cold? Do you feel pressure? Things like that. There are other neurons largely inside our brain that process these signals, kind of make sense of them. And there's other neurons that are involved in sending the signals to the muscles to cause us to move, to run, to jump, to dunk a basketball, to catch a football, things like that. So this is what the neuron looks like. It's this kind of alien looking thing. Over here, you see the inputs coming from outside the neuron into these kind of finger-like appendages that are looking to kind of pick up certain inputs. And neurons are waiting for certain kind of keys to open this lock. Some of these things floating around here we will talk about on the next few slides, but some of those inputs are responsible for feeling good or feeling addicted or feeling close, attached to other people. So anyway, when there's enough of these kind of inputs, the neuron kind of picks them up and houses them. And then when there's a sufficient enough of that input, then it relays that message, kind of processes it through this long kind of highway into the outputs where it gets spit out and it gets picked up by another neuron with its kind of finger-like appendages to relay that message. And this happens incredibly fast. But this is the anatomy, what that neuron kind of looks like. So think of it as kind of getting the input, processing that message along the way, and then spitting it back out at the very end as a sort of output for other neurons to pick up. So as I alluded to earlier, there are 3 different types of neurons. And here's some formal language for you guys. The first type of neuron is a sensory neuron. So these sensory neurons respond to stimuli from the outside world that's picked up through our sensory organs. So these sensory neurons are in your hands, they're in your feet, they're in your lips, they're throughout your body, and they allow us to feel things like feel like this is hot or cold. There are sensory neurons on your tongue, right, connected to your taste buds. Say, hey, this tastes salty, right? That sensory neuron is firing when it gets enough of that input of salt from the outside world. It's firing in your fingertips when it feels that sort of pressure, like, hey, someone's holding my hand right now. There's sensory neurons in your eyeballs, right, that are responding to different wavelengths of light that let you know, hey, I see something. Those sensory neurons are just responding for kind of picking up sensations from the outside world. Those sensory neurons are picking up information from the outside world. Motor neurons are the second type of neuron. Motor neurons are in charge of kind of moving our muscles. So when you decide, hey, I need to run really fast and get away from this cheetah, or I need to jump really high so I can, run and jump and catch this ball, for instance. The way that you are moving your body is through motor neurons. So again, these are neurons that are connected to these muscles. And when those neurons kind of fire or process that message, then the muscles kind of contract and cause you to run or jump, things like that. And the third type of neuron that is the most common is the interneuron, or just the connector neurons between sensory neurons and motor neurons. So I mentioned earlier that our sensory neurons are throughout our body. Those are attached to our sensory organs to feel, to hear, to see, things like that. Then once they go from that sensory neuron, then they go up the spinal cord, right? And that sort of process is through interneurons. They get to the brain, which is just a bunch of interneurons that are relaying this message between the different lobes and parts of the brain, so we can process it and make sense of what's going on here. And then at the very end, after we realize like, hey, I'm feeling something, I'm feeling someone hold my hand, but I look around and no one's there. I don't know anyone here. This is kind of weird. I need to pull my hand away. Then these interneurons would relay that message to a motor neuron that would fire and cause your muscles to kind of retract and kind of pull your hand away. What's wild is your brain is full of just interneurons, but no sensory neurons. So if you were to cut someone's head open and like kind of touch their brain in there, they wouldn't be able to feel it, right? Because there's no sensory neurons in your brain. It's just these interneurons that are the most common, the most prevalent throughout our bodies. And they're just the kind of relays between sensory and motor neurons. So earlier when I was talking about the kind of anatomy or structure of a neuron, I talked about the inputs, how they come from other neurons, other cells, and then they get picked up through these finger-like appendages, and then they get processed throughout the neuron, and they get spit out at the output at the very end. Each part of the neuron has a specific name. Those finger-like appendages are actually called dendrites. So dendrites, think of it as just finger-like appendages kind of floating out there, waiting to pick up inputs from other cells. And on the next few slides, I'll talk about what those inputs are. But right now, just think about dendrites as these kind of finger-like appendages kind of reaching out, waiting to input this message. They're the locks looking for the keys out there. So then once the dendrites receive enough of this input, then they bring that message along into the soma, or the cell body, where it's housed, where they're kind of waiting for enough of that message. And then when they receive enough of it, they're like, okay, great, we need to send this message on down. That message gets relayed or processed through this really long highway referred to as the axon. The axon is a way of just kind of like relaying messages really, really quickly until it reaches the very end, or the output. And those sort of knobs at the end are referred to as terminal buttons. Once the message gets to the end, then they kind of leak out and they get picked up by the next neurons and dendrites. If these axons are the kind of like super highway that allows these messages to get relayed and sent from one neuron to another really quickly, myelin sheath is this sort of fatty substance that surrounds the axon, kind of like these little hot dog buns. And what the myelin sheath does is it speeds up that whole process even faster. So there's some neurons that we use all the time, right? When you're learning how to walk, for instance, you're using those motor neurons that you had never used before. But then once you get your balance, you start walking and then running, you start, going on hikes over and over again. Those motor neurons in your leg, those keep firing over and over and over again. Your body says, hey, we keep using this neuron. Let's add some fatty sheaths to it. Let's add this myelin sheath that helps us process that message even faster. The same thing is happening in your brain, right? When you're thinking, there are certain things that you think about over and over and over again, like your boyfriend or girlfriend, or your siblings, or your parents, or your favorite TV show. The first time that you see this person, for instance, they're brand new. You've never seen this person before. You have parts of your brain, right, that light up, neurons that are firing when you see what this person's face looks like. Other parts of your brain that are lighting up, neurons firing when you hear what their name is. And then once you start talking to this person, using their name, seeing them over and over and over again, day after day, week after week, month after month, year after year that you've known this person, those neurons are being used over and over and over again. Every time you see this person, every time you hear their name, every time you bring up that memory of what they were like, those same neurons are firing. So to help that neural pathway speed up, get used over and over and over, because it's being used so frequently, your body will start adding myelin sheath to it. It's just a fancy way of letting that neuron process information faster. The same things happens when you study, right? When you study, you're thinking about neurons and psychology, and you're hearing my voice. So you're probably thinking about Collison, you're thinking psychology, you're thinking neurons. And then you might already have other memories in your brain stored from biology, where you talked about neurons with your old, bio teacher back in high school. Those memories are stored, right? So then when you're hearing this new terms about dendrites and soma and axons and things like that, and you're hearing my voice and connecting it to psychology, your brain is literally kind of moving those neurons closer together so they can relay that message, because you're hearing about it, you're seeing it in slides. Those same neural pathways will be activated later tonight when you read this in your textbook. When you study it, when you get to the study guide next week, those same neural pathways are going to be used over and over and over again. So when you're sleeping tonight, your brain is kind of taking stock of which neurons are being used a lot. And if you keep connecting all these neurons that are firing when you're learning about psychology and bio and dendrites, your brain will literally start adding myelination, right? It'll start adding this myelin sheath to those neurons that allows you to recall the information even faster, right? To think and recall this information quicker than you were before, which is why it's so important to study and to sleep well. Make sure that you're getting enough sleep and not just using cramming sessions for your exams, because when you sleep, your brain is literally changing. Those neurons are starting to move together in ways that they can fire quickly. And when those neurons are getting used over and over again, your brain will even start adding myelination to them, this myelin sheath that allows you to think faster, to move faster. Just cool. So how does a neuron really fire? I keep using that term, right? How does it release one message and send it on to the next? That process is referred to as neural activation or firing. And right now, your neurons aren't doing anything unless they need to. They're at rest. There's chilling there. And this sort of resting potential is both an electrical and a chemical process that causes them to fire. So right now they're hanging out, more negative ions in the inside, more positive ions on the outside. And what happens when those dendrites are just kind of reaching out, searching for that key, just waiting for it, is when it receives the right input, the key goes into the lock, gets inputted, and basically through these chemical and electrical process, your resting potential will start opening these kind of what's referred to as sodium potassium pumps, and it'll literally start kind of changing this plus and negative throughout the cells that allows that message to be relayed through the dendrite, through the soma, and all along that axon until it reaches the terminal buttons where it can release that message, and then the whole process starts over again. So your neurons aren't firing unless they receive the right input. They're at a resting potential, and then when they receive the right sort of key that they're looking for, the threshold will be exceeded, and then they fire. Neurons fire in an all or none response, right? They fire in an all or none response, meaning they're waiting, then they get enough of it, and then they fire, right? They pass that message on down super quickly, fraction of a second, and then it gets released, and then the process starts over and over again, which is pretty incredible. Every thought we have, every perception that we come up with when we're watching TV, when we're talking to friends, when we're studying, when we're driving, like our brains are going nuts, just making sense of the world around us, which allows us to think, right? It develops a sense of consciousness that makes us uniquely us, which is incredible at how this all works. So I mentioned earlier that the neurons that we use over and over and over again will add this sort of myelin sheath to it. Think of it as just this sort of insulator that allows messages to be processed even more quickly throughout those neurons. Every now and then, some people will develop diseases referred to as multiple sclerosis, where the body will start kind of turning on its myelin sheath. It'll think it's a foreign substance. It'll start attacking its own myelination, even though it shouldn't. And what happens is those motor neurons that someone was using to walk and talk and move their arms, all of a sudden, they'll start being a little bit trembly. The balance will start to go as one of the kind of warning signs that, hey, those neurons aren't working the way that they're supposed to. Or kind of memory loss. It just kind of takes harder to bring up some of those memories that used to be really, really quick. That is an example of the sort of damaged myelin sheath that's common in multiple sclerosis. So if you know someone with multiple sclerosis, it's basically this sort of autoimmune disease where their body is starting to turn on their myelination. So balance, tingling sensations, like all of those sort of neurological symptoms can be tracked back down to this multiple sclerosis that's damaging their myelin sheath. So how does one neuron cause other neurons to fire into this kind of like perfect storm, this sort of spreading activation throughout people's brains that allow us to make sense of incredibly dynamic and complex worlds? The answer is through neurotransmitters. I kept using the term earlier, like the inputs, right? Those dendrites were hanging out there kind of waiting for the right signal, the right keys, right, to unlock the locks. That input that I was referring to are referred to as neurotransmitters. And there are a lot of different neurotransmitters out there. Here are some examples of them, right? These little kind of dots out there kind of hanging out in between the neurons. Some of the neurotransmitters that you might have heard of before are things like serotonin. Serotonin, the kind of feel-good neurotransmitter. If you have enough serotonin coursing through your brain, that neurotransmitter is sending messages saying, I feel good. I feel happy, I feel good, I feel content. And if someone doesn't have enough serotonin coursing through their brain, they're not relaying that message. So instead they're feeling like, I feel kind of bad, I feel kind of sad, I don't feel content, I don't feel good enough. So serotonin, right, linked to depression. And it can be explained by the neurotransmitter not having enough of serotonin. There's other neurotransmitters out there that you might have heard about, like dopamine. Right? Dopamine, like this is rewarding. Let's do this over and over and over again. So when someone is addicted to drugs and alcohol, addicted to gambling, that dopamine is going nuts, right? There's so much of it in the dopamine reward pathways that dopamine just kind of coursing saying, this is really addictive. I want more and more and more. Other neurotransmitters out there like oxytocin. Oxytocin coursing through your brain, letting you know that, hey, I feel attached. I feel attached to this person. I love them, right? I'm sad when they go. I'm happy when they return. I'm attached to this cute puppy dog, right? The same way like your cute puppy dog has oxytocin in their brain. It's sad when you go, it's happy when you return, right? Forming that sort of attachment. There are neurotransmitters related to like energy and things like acetylcholine that's involved with like movement and activating muscles. So if someone doesn't have enough of that, they might be kind of paralyzed, right? If they're bitten by a poisonous spider, for instance, that's preventing that neurotransmitter from being passed on from neuron to neuron. They can't move their arms, for instance, if they're bitten on their finger by a spider, for instance. But anyway, neurotransmitters are basically just these sort of like kind of messages, these inputs that get relayed from neuron to neuron. And this all happens incredibly quick. So what's going on is you have these dendrites kind of reaching out, waiting for those right neurotransmitters to get kind of picked up. And then the neurotransmitters get brought in through the dendrites and they get housed in the soma or the cell body. So when there's enough of those neurotransmitters, the soma kind of packages them up into a kind of vesicle. A vesicle is just a fancy word for just like a little car, basically. It piles them all in, buckles the seatbelts, and then the vesicle, when the neuron fires, right, its threshold is exceeded, it's received, all the spots are filled, it's exceeded, enough to fire. Then that vesicle gets shot through the soma into the axon, and it goes all the way down the axon until it reaches the terminal buttons way down here. And those vesicles reach the edge, the terminal button, and then they kind of spill out into this gap. The gaps between neurons are referred to as kind of synapses. And this gap between one neurotransmitter's terminal buttons and the next dendrites down here, kind of finger-like appendages waiting for those right neurons, this gap specifically between a terminal button and a dendrite is referred to as a synaptic cleft. And what you'll see here is when the vesicles, right, those little cars with the little neurotransmitters inside, they get to the edge, notice that they spill open and all these neurotransmitters are now falling out into the synaptic cleft where they're hanging out. And hopefully the dendrite for the next neuron has its finger-like appendages and it's waiting to pick them back up, put them in vesicles, and then fire and send that message on down. But what if all of those neurotransmitters don't get picked up by the next dendrite? What happens sometimes is they might get kind of floating off, and get kind of cleaned up by other kind of glial cells. That's job is just to kind of clean up all of these neurotransmitters that aren't being used. Or what can sometimes happen is if they're hanging out in this gap for long enough, they aren't getting picked up by the dendrite, they might get kind of reabsorbed by that terminal button, right? And then when the next vesicle comes down, they'll get re-released again. So they have another chance of being spilled out and then picked up by the dendrite again. So that's a lot, but one way of thinking of it, that's those vesicles housing the neurotransmitters going through the axon into the terminal button, reaching the edge, opening up all those neurotransmitters, floating out into the synaptic cleft, right, the synapse between neurons. And then the next dendrite over here is kind of reaching out and waiting to let all of its, keys in if they fit the right lock. So it's looking specifically for certain types of neurotransmitters, and then it receives enough of them. It'll package them all up into vesicles, fire, there's all or none response, and relay, kind of rocket all of those neurotransmitters through the axon to the terminal button where they will be released again. And that process happens over and over and over again. And this is kind of what it looks like, right? You see the terminal button up here, you see the vesicles, those little circles, like the big circles, kind of cars that are housing all of those neurotransmitters. When they reach the edge, they spill out. A lot of those neurotransmitters are then being picked up by the next dendrite, causing them to fire, but not all of them. Some of them are kind of floating out here. They'll either get kind of cleaned up by glial cells or they'll get reabsorbed by the previous terminal button. And that's how your brain works, right? It's these neurons firing over and over and over again. When we see pictures of the brain kind of lighting up, what's happening is we're kind of detecting which neurons are firing in the brain, what sort of activity is going on. And then when people take drugs, right, when they drink coffee, for instance, it changes the way their brain functions. When people take cocaine, for instance, a stimulant, it changes the way their brain functions. When they drink alcohol, right, a drug that's a depressant, changes the way their brain functions. When they get bitten by a poisonous snake a venomous snake, or they eat a poisonous mushroom, right? those sort of drugs will change the way the neurons fire. And depending on the type of drug that they're exposed to, it may help or hurt how that neurotransmitter, it'll either help or hurt that neurotransmitter as it's doing its job. So anyway, there are some drugs out there that act as agonists. Basically, they're kind of mimicking the effects of a neurotransmitter to help it do its job better. So there are certain drugs out there like cocaine, for instance. And what happens when you do a line of cocaine, for instance, it activates those sensory neurons, it gets kind of picked up. That cocaine from a like chemical perspective really resembles a lot of like acetylcholine. It acts as a stimulant. So all of those sort of things of like, this is addictive, this is fun, let's start rushing, let's start like thinking faster, let's start moving. All of the extra cocaine that you took is basically kind of like a fake key. It's being picked up by those dendrites thinking that it's like, that it's energy, that's movement, that people should be moving and thinking and doing things faster as a stimulant, which causes people's like heart to race, causes their thoughts to race, causes them to start moving around and being a lot more energetic than they were before. There are some drugs out there that act as antagonists. Antagonists, meaning that they are kind of blocking that neurotransmitter from doing its job. So there are some neurotransmitters in your brain right now, like GABA, for instance, that are being picked up and saying, let's cool things down. Let's slow things down. Let's relax. Let's fall asleep. You're feeling tired. When those neurotransmitters are being picked up, you start winding down. So when you drink coffee, right, caffeine, for instance, you do a Red Bull energy drink and things like that. What happens is the neurotransmitters in that product like make their way into your brain, right? And your brain starts blocking those sort of GABA receptors from saying, let's slow things down. So they block that, let's slow things down, let's be tired, let's fall asleep. So instead, your brain isn't saying that. It's saying, let's be awake, let's do stuff, let's be energetic instead. I mentioned earlier, if you got bitten by a spider, venomous snake, or something like that, you might be kind of paralyzed. You might not be able to move your arm, for instance. What's happening is that sort of venom, it's being picked up by your body, right? It's being picked up by your nervous system. Those neurotransmitters are acting as antagonists against acetylcholine. That neurotransmitter said, let's move, right? When that neurotransmitter is being picked up, it's being released to those motor neurons, you're able to like contract that muscle and walk and talk and move your arm around. But if you've been kind of bit by a venomous snake, for instance, that venom is in your arm and it's acting as an antagonist for acetylcholine. So acetylcholine is trying to work. It's saying, hey, move, 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 but you can't, right? It's blocking on the dendrite. That sort of venom is kind of covering, kind of blocking acetylcholine from being picked up. So even though you're trying to move, you literally can't. So anyway, these are all ways of how kind of drugs affect the brain. Sometimes they help and sometimes they hurt those neurotransmitters from doing their job. If you were depressed, for instance, you could take Prozac, Zoloft, Paxil, right? Other types of antidepressants, right? And the way those antidepressants work is they're drugs. Specifically, they are selective serotonin reuptake inhibitors, SSRIs for short, selective serotonin reuptake inhibitors. If serotonin is that neurotransmitter that's saying like, I feel good, I feel good, I feel good, what we want to do is act as an agonist and help serotonin do its job better. So this person is feeling better, right? They aren't feeling as depressed. So what happens is when you take a selective serotonin reuptake inhibitor, what this drug will do is it'll sit in the synaptic cleft, right? The synapse between neurons, And what happens is when serotonin gets traveled along in these vesicles, reaches the end, it starts spilling out. Hopefully it gets picked up by the next dendrite, right? So we can relay that message and say, yep, I'm feeling good. Yep, I'm feeling good. I'm feeling good. I'm feeling good. But for people that are clinically depressed, sometimes serotonin isn't working well. Maybe they don't have enough of it. So sometimes that serotonin might kind of float away and get cleaned up by glial cells. Or it might be so quickly kind of reuptake or kind of reabsorbed by the terminal button, it never has a chance of making it down to the dendrite. So what happens is when you take Prozac or Zoloft or Paxil, any of these SSRIs, what they're doing is selectively helping serotonin be inhibited on the reuptake. So selective serotonin reuptake inhibitor. They're basically acting as kind of walls up here, blocking serotonin from being reabsorbed. So when this serotonin tries to bounce off this wall, it's getting rejected. So it's hanging out in the synaptic cleft for longer periods of time, which makes it more likely to get a chance to be absorbed by the next dendrite, which fires and says, yep, I'm feeling good. So when you take Prozac, Zoloft, Paxil, right, any of these SSRIs, they're acting as agonists, helping serotonin do its job better. So why should we care about these? Why should we care about neurons and drugs and things like that? Is because there are a lot of kind of neurological disorders, right? There's problems with people's brains and how they're working. Specifically, sometimes those neurons aren't working the way that they should. If someone has Alzheimer's, for instance, there might be plaques forming between the neurons that they can't relay the message. So all of a sudden, they don't know where they put their keys. They don't know the names of their kids. They don't know where they live, right? Because they can't access that information because those neurons aren't able to kind of relay the message like they should. Sometimes the information inside the neuron is getting kind of tangled. So those neurons die and they can't relay that message anymore of like how to walk, how to talk, how to breathe. And eventually Alzheimer's Alzheimer's unfortunately is degenerative, meaning it's getting worse over time. The plaques and the tangles, they start adding up, the neurons start to die, and the person's memory goes from bad to worse. And eventually those sort of motor neurons of not just thinking, but also breathing will start to fail, and people will eventually die from Alzheimer's in the more advanced stages. When people have Parkinson's disease, right, those sort of involuntary muscle spasms, it's related to dopamine, right? An insufficient amount of dopamine in certain parts of the brain. So neuropsychologists will focus on inventing different types of drugs. Things like L-Dopa, right, that acts as an agonist, helping create more dopamine in certain parts of the brain so people don't have those same sort of kind of muscle spasms over time. You know, like people like Fox and folks like Muhammad Ali have. So anyway, why should we care? First of all, our brain is going nuts processing the world. It's important to know how it works. But also, we often kind of take it for granted until our brain isn't working the way it's supposed to, until our neurons aren't relaying messages the way that they're supposed to. And then the more we understand about what's going on, then we can invent drugs and therapies, for instance, that will change how neurons work. So far, we've been talking a lot about neurons, and there are neurons throughout your entire body. Those neurons are just referred to as the nervous system, right? Those 100 billion neurons throughout your body. And if you wanted to break it down even further, you can say, well, some of those neurons are part of the central nervous system because they're in your brain and they're in your spinal cord, right? Centralized nervous system. There are other neurons that reach throughout your body, your fingertips to your legs, for instance. Those are part of your peripheries or the peripheral nervous system. And if we wanted to break that down even further, we could talk about autonomic and sensory somatic aspects of your peripheral nervous system. So if you are a visual person, you can think of all the neurons in your body. Some of them are central nervous system, right? Part of your brain and your spinal cord, really, really important. Also, you have your peripheral nervous system, so the neurons throughout your body. Some of those are just sensory neurons connected to your eyeballs, connected to the bones in your ear, right, that are being vibrated when you hear things. Other sensory neurons in your fingers, like feeling things like, is this hot or cold? Do I feel temperature? Do I feel pressure? There are other neurons connected to your muscles, right? Those motor neurons. Some of those motor neurons are part of the somatic nervous system, meaning we have control over them. Meaning if you think about, hey, I want to pick up my foot, you can do it. I want to put my foot down, you can do it. If I want to put my hands a certain way so I can hold a coffee cup, for instance, or you want to move your fingers a certain way and flip someone the bird, like that's all part of your voluntary control, somatic nervous system. There are other neurons connected to muscles that are on their own. They're part of this autonomic nervous system. They are firing without you even knowing that they're firing. Like breathing, for instance, moving the muscles related to your lungs to let more or less air in. Your heart being a muscle, kind of pumping that. That's all part of your autonomic nervous system. And that autonomic nervous system, if you even want to get more nuance, can be broken down into a sympathetic versus a parasympathetic division. And it's largely dictated by, are you stressed? Are you experiencing like stressors in your environment? Like, are you walking around and then all of a sudden there's like a bear in your living room? Do you imagine like watching TV and all of a sudden like a bear jumps out and it's like snarling and it's big and it's about to eat you? What's going to happen is automatically, right, this autonomic nervous system, your sympathetic nervous system is going to kick on saying, we need to fight or flight. We need to kill this bear, right, defend ourselves, or we need to run out that front door as fast as we possibly can. So what happens is automatically, without you even thinking about it, your body starts diverting all of its energy to those motor neurons that are going to help the muscles in your legs run as fast as they can, help the muscles in your arms like be as strong as they possibly can to defend yourself. And the other things like the neurons connected to your stomach that's saying, hey, let's digest this food. The neurons connected to like your thyroid and your hormones saying, hey, let's go through puberty, let's start menstruating, like all of those sort of things, like processing your food, going through puberty, like they're important, but they're not as important if you like don't fight off that bear right now. They're important to get to, right, to live healthy, to develop into the person that you're supposed to be. But who cares if you digest this food right now if you're dead because the bear ate you? So instead, what you find is that the sympathetic nervous system will kick on when you're in fight or flight situations. And then as soon as you like outrun the bear, then your body relaxes into this parasympathetic nervous system. It's like, all right, let's rest and digest. Let's start processing, let's food, let's start going through puberty, let's start like regulating our body the way we're supposed to. Notice that the sympathetic and the parasympathetic both cannot act at the exact same time. It's one or the other, which is why like when people are really like stressed for finals and things like that, they might start getting sick because their body wasn't repairing itself. Or when you're really stressed out, you might have like GI issues and like stomachs upset, things like that, because you weren't like processing that food the way that you normally do. It's important to kind of relax and let your parasympathetic nervous system kind of kick in. So the reason why I'm talking about, talking about all of this, I went on a tangent real quick, for a bit. Let's briefly talk about the peripheral nervous system, but I actually want to spend a whole lot more time talking about your central nervous system, specifically the brain. But first, real quick, peripheral nervous system, think of it as autonomic is automatic, not under your conscious control. And part of that autonomic nervous system is either the sympathetic or the parasympathetic. Fight or flight versus let your body kind of rest and digest. The sensory somatic nervous system is the motor neurons. I want to move this. I want to hear my phone go off and then react to it, and pick it up, see who it is, hit the button to say answer or not, things like that. That's it for the peripheral nervous system. I mentioned I want to talk a whole lot more talking about the brain, part of the central nervous system. And this is what your brain looks like. This is the side portrait. So the person is actually kind of looking towards the left over here. Like this would be their nose over here. This would be their mouth, for instance, their eye. And this would be the back of their head over here. So the same thing here, the way that the person is holding the brain, this is the forehead right here. This is where the eyes would be. This is where the nose would be. This is where the mouth would be. And the back of their head would be way over here. So what's really interesting about this kind of 8-pound organ is if you were to cut the skin on someone's head and kind of pull it back, you would see that bone, right? The kind of cranium. If you took a bone saw and then kind of, opened it up, what you would see is the brain, right? This kind of lasagna looking jello, kind of gelatinous substance. And if you were to kind of reach down in there and kind of touch it, first of all, the person in there wouldn't be able to feel it. They would feel when you cut the skin, right? Because there's sensory neurons in your skin. But once you pulled the skin back and kind of cut the bone, if you actually touched this part of someone's brain, they wouldn't be able to feel it, right? Because this is all interneurons. There's no sensory neurons in there. But the first thing that you would notice is on the outside, there's kind of like this like saran wrap on it, this kind of cellophane kind of thing. This is referred to as the meninges. You can kind of see it's a little shiny over here, right? This kind of covering. The meninges, this kind of outer covering, does a nice job of just kind of like holding it all together. All this cerebral spinal fluid in there, all the neurotransmitters that kind of floating around neurons, the meninges kind of do a nice job of holding all of that together. And when people experience like, what's the word? When people get meningitis, sort of like inflammation, infection of the meninges, right? This sort of saran wrap covering the brain. It's a really big deal, right? Because it's so important for how people function. So anyway, meninges are just the kind of outer covering. Once you cut that, kind of peel it back, then what you'll notice is that the brain has these sort of like lasagna-like ridges on it, right? These sort of like high pieces and low pieces. The outside portion of the brain, right? The sort of lasagna looking thing that you would actually touch is referred to as the cerebral cortex. Right? Cerebral cortex is just the outside portion of someone's brain. And it is comprised of the sulci and gyri. Think of sulci as these kind of sunken in ridges and the gyri as these kind of high ridges, right? So the sulci kind of sunken in pieces in between, and the gyri, these kind of high kind of ridges that look like lasagna. So the cerebral cortex can actually be broken up into two different cerebral, meaning brain, cerebral hemispheres, one on the right side and one on the left side. And I'm saying right side over here, because if you were to put yourself in this person's position, right? This is their brain, this is their forehead, this is their nose, this is their eyes over here. If you were to put yourself in their position, this would be the right side of their head. This would be the left side of their head. So this is called the right cerebral hemisphere and the left cerebral hemisphere. So your brain is basically two halves, and it's connected in the very middle here through this large bundle of neurons referred to as the corpus callosum. So the corpus callosum is just a thick bundle of neurons that allows the right cerebral hemisphere to communicate with the left cerebral hemisphere. So there's this kind of wives' tale out there that people only use 10% of their brain. No, not at all. That is completely false. That would be so evolutionarily unadaptive. Like why would people do that? Instead, people use 100% of their brain, right? People use all of it, their right and their left cerebral hemisphere, all of it. There's wives still out there that some people are right brain versus left brain, right? Some people are more creative, some people more analytical. No, people are using both parts of their brain all of the time. In fact, the memories from one side can be accessed by the other side through that corpus callosum, that bundle of neurons in between. And I'll show you a cool picture of what that looks like in a second here. But first, notice the human brain. Check out the corpus, check out the cerebral cortex, right? The outside, how it has all the sulci and gyri, all those kind of ridges to it. So different than like a songbird. This is the songbird's forehead. Notice that it's really smooth as compared to the humans. Very few sulci and gyri. And that's because humans are a whole lot smarter than songbirds are. The reason why human brains are so wrinkled like that with sulci and gyri is it increases the square footage, right? It increases the amount of space and the amount of connections neurons can access each other. So your ability to be, smart, to think, is just, can these neurons communicate with each other? Can they access each other really quickly to, see something, think about it, name it, process it, predict it, what's going to happen? And by having these sulci and gyri in here, it gives us so much more opportunity, so many different combinations for those 100 billion neurons to connect with one another, to see something for the first time, give it a name, and then predict what's going to happen next. So you see the human over here, you see monkeys start to have more sulci and gyri, not nearly to the extent as humans, but monkeys are a whole lot smarter than a bird or even a mouse. And notice that certain parts of the brain, what are referred to as lobes of the brain, are a little bit different with each animal. The mouse, check out the side of its brain is huge. The songbird, the side of its brain is huge, right? That lobe is devoted largely to sound, to hearing things, right? They have really big ears. They, chirp and communicate via, tweets and chirps and songs and stuff like that. Monkeys are a lot more intelligent, a lot more sulci and gyri. The front part of its brain, right? If this is the monkey's eyes and its nose over here, the front part of its brain isn't quite as advanced as humans are, right? And that's probably because like monkeys don't have like the same richness of personality. They don't have the same emotions. They don't have the same depth of thinking and anticipating consequences for your actions the way humans do. But anyway, they certainly do have some, a lot more than a mouse did or even a songbird did. So kind of cool. Here's a picture of your brain cut in half. So if we have the right cerebral hemisphere and the left cerebral hemisphere, I mentioned earlier that corpus callosum is this kind of bundle of neurons, right, connecting the right and the left hemisphere. If you were to kind of turn sideways and see this person's forehead, their eyes, their nose, their mouth over here, what you would see is their left cerebral hemisphere is being connected right through this bundle of neurons called the corpus callosum. And what's so wild is that this corpus callosum connects the right and left cerebral hemisphere. And when people have like grand mal seizures, right? If someone is epileptic, for instance, and their body starts convulsing, they start having a seizure, all the neurons in their body are firing uncontrollably, right? Their muscles are moving, right? They're seizing when they're not supposed to. That can spread into the brain as well. So if someone has a grand mal seizure, what you can do is literally cut their corpus callosum in half. So when the right part of the brain starts seizing, right, all the neurons start firing on the right side, they can't access the left side. So it basically prevents this person from having a grand mal seizure. And what's so wild is people's brains are incredibly plastic, right? They have neuroplasticity, meaning that if you were to damage someone's brain by cutting their corpus callosum in half, they're still going to survive. They can still like relearn. They can relearn how to think, how to talk, how to access like memories and things like that. It just takes time. There are other neurons in their brain have to start moving around. They have to start shifting and like start communicating in different pathways to get access to that information. This is the same way as if someone had a stroke, for instance, and like part of their neurons all died because they didn't have enough oxygen. They might, forget words, they might forget how to talk or forget how to walk. Through therapy, depending on the severity of the stroke, they may be able to regain functioning. They may relearn that person's name. They may relearn how to talk and how to walk. Basically, other neurons in other parts of the brain will start kind of making up for it, which is just incredible that, that our brains do this. So anyway, I've been talking a lot about the brains. Let me talk about the different lobes of the brain. So it is true that different parts of your brain do different things. Way back in the day, there was this pseudoscience, this kind of fake science out there, where they would go around and measure people's heads. It was called phrenology. And what they would do is say, oh, there's a bump here. That means that you're really funny. Or there's kind of a dent over here. That means that you are really argumentative. That's complete fake. It's not real. It's pseudoscience. There's no way that like the bumps on your skull, on your cranium, tell anything about your personality, right? So they were wrong about that. This was a racket where they would charge people to measure their brains and then tell them about who they are. This is fake, but they were onto something in phrenology that different parts of your brain have generally similar functions. And there are 4 different lobes of your brain. The first lobe is referred to as the frontal lobe. The frontal lobe, think of it as just right here, right behind your forehead. This is really advanced in humans. And that's because your frontal lobe houses your personality. It houses things like your emotions. How do you feel about that? Your ability to like reason, to think. Are you impulsive or not? If I were to like take ice cream and put like cough syrup and sriracha and garlic and pickles in it, and I turned it into this like pickle, garlic, sriracha ice cream, you would look at that and be like, ew, no, I'm not tasting that. I know it's not going to taste good. That's all your frontal lobe. You can anticipate the consequences of your actions. You can look at something and anticipate what's going to happen. What's it going to taste like? All of that is your frontal lobe because it's so advanced. Your dog, for instance, a monkey at the zoo, is going to taste that ice cream before it knows it's really bad. You don't have to. Your frontal lobe that houses all of that, like personality, reasoning, emotions, anticipating consequences of your actions, your frontal lobe isn't fully developed until people are about 25 years of age. So it's kind of wild that like people are 18 and we're saying like, all right, like, make decisions about what you want to do for the rest of your life. Do you want to join the army? Do you want to go to college? When they're 21, like, hey, anticipate the consequences of your actions. Drinking and driving, like, is this going to be a good idea? No. Deciding who you want to marry, who you want to live the rest of your life with. Your personality, right, is still changing. Your frontal lobe is not yet developed. And one of the ways that they knew that your frontal lobe is so key to who you are, emotions, personality, things like that, impulsivity, is by this poor guy named Phineas Gage. Phineas Gage had a rough life. He worked the railroad, the transcontinental railroad from the East Coast to the West Coast, right? In California, there was a gold rush, and they realized, hey, let's start like, this westward expansion. People are going to need ways to go from the East Coast to the West Coast. Let's build a railroad. So what they would literally have to do is, take these kind of railroad, ties, kind of, lay them out, and then take the ties and kind of like connect them to the ground. So when the train goes over it, the railroad doesn't move. So one of that job is other people would kind of carry the track, and other people would carry these railroad ties like Phineas Gage. And what his job was kind of laying the spike down in there and then taking this real big hammer and kind of slamming it down and basically kind of sticking the railroad tie into the ground. And they would do this, they'd keep walking until they reached all the way from the East Coast to the West Coast, which is tough, right? Because railroads need to go in straight lines. And I don't know about you, but it's hard to walk in a straight line because there's mountains and trees and boulders and all sorts of streams, all sorts of things out there that make it really difficult to make a train go in a straight line. So other folks in the railroad would carry around dynamite. So if there's a big boulder, for instance, and they need to lay the track right there, what they would do is light the dynamite, blow up the boulder, and then folks like Phineas Gage after would come along, lay the track down with his spike and kind of nail it in there. So anyway, Phineas Gage was a really like happy guy. He was always goofing around. He was really joyful. He was really religious. He would talk about God. Like people just loved being around Phineas Gage. Then one day, someone laid down the railroad track. He comes along with his spike, sets it down. Phineas Gage didn't realize is that there's actually dynamite underneath it. There was dynamite underneath this track. So when Phineas Gage comes along with his spike and his hammer, he slams down on the hammer and blows up this dynamite. And the rod that he was carrying shoots back out, right? It exploded from the dynamite and it goes right through Phineas Gage's eye, right through his eyeball and destroys his frontal lobe back here. So all of Phineas Gage is like, there's blood, there's guts, like it's disgusting. All of Phineas Gage's friends think that he's dead, right? He literally has a tie through his brain, blood everywhere. They rush him to the doctor, not thinking Phineas Gage is going to live. But he did. He survived. And what they found is by damaging his frontal lobe right here, he wasn't the same person that he was afterwards. He could still walk, he could still talk, he could do all of those things. But now he's really angry. He was bitter. He cussed a lot. He drank a lot. He didn't go to church. He got in a lot of fights with his friends. Like, just wasn't really the same person. And that's because you damaged someone's frontal lobe that houses their personality. They aren't going to be the same person that they were before. The same rationale for like frontal lobe lobotomies. Back in the day, before people really understood about ways of changing personality and things like that, mood and emotions. Sometimes if people had psychological disorders, they would stick a kind of metal rod up someone's nose and kind of lesion or scratch their frontal lobe, and it changed their personality. If they were really impulsive and psychopathic, it made them less impulsive, psychopathic. If someone was really moody, it changed their mood, right? That's because you literally scarred or damaged someone's frontal lobe. Luckily today, we aren't doing frontal lobe lobotomies. But the reason why I'm telling these really gross stories is to highlight your frontal lobe houses your personality. But there are other parts of your lobe. The very back part of your head is referred to as the occipital lobe. Your occipital lobe, largely responsible for vision. So when you see something, right, you have these sensory neurons in your eyeballs, then they have these kind of interneurons that connect all the way to your occipital lobe way in the back. So when you see someone's face, the back of your lobe starts lighting up. If you get in a car accident or someone hits you in the back of the head with a chair, I don't know why you're like a WWE wrestler or something, but like someone sneaks up behind you and hits you in the back of the head with a chair, you might start seeing stars. What happened is you just damaged your occipital lobe. That brunt impact caused those neurons to start firing. You start seeing things that aren't really there, right? You start hallucinating and start seeing stars. Other parts of your brain on the side over here is your temporal lobe, right next to your ears. And no wonder, right? Your temporal lobe does things to comprehend language, make sense of words, coming up with the right word, how to express yourself, how to understand what other people are sharing, kind of things like that, kind of temporal lobe. Makes sense that that rat's brain that we showed you earlier had a huge temporal lobe, right? Because they're really great at hearing things like, is someone coming? What's going on? Is that the sound of food? What is it? humans' temporal lobes aren't nearly as advanced because our hearing isn't as great as other animals. And the fourth part of your brain is behind your frontal lobe, above your occipital lobe, over here, this little region. It's called the parietal lobe. And your parietal lobe is really good at sort of like attention and like, where is my body in relation to itself? When I'm reaching for something, how far do I have to reach? When I'm drinking a cup of coffee, how close do I have to get it to my lips before I can actually take a sip? I don't spill it on me. All of that sort of like making sense of the world around you is largely processed via your parietal lobe. In fact, there's a couple strips right here at the front, right behind the frontal lobe, right in your parietal lobe. These strips are really kind of like devoted to processing information from your sensory neurons on your lips, on your hands, on your elbows, and different parts of our bodies are more sensitive than others, right? That's because in our parietal lobe, these strips, certain parts of our body kind of chew up more kind of brain space. Like your lips are far more sensitive than your elbow is. That's partly because we have a lot more of these space chewed up in our parietal lobe, right, to kind of feel like our eyebrows, our face, and our lips and things like that, but very little for like our knee, for instance. Our knees and our elbows just aren't nearly as sensitive as our fingertips, for instance. These sort of strips and this sort of diagram that kind of represents like the size of the human body based on how sensitive it is in the parietal lobe is often referred to as the homunculus. You know, so big faces, big eyes, small elbows, big hands, things like that, just because those parts of our bodies are so much more sensitive than others. What else? I mentioned our cerebral hemispheres, our right and the left cerebral hemisphere, and that corpus callosum, right, that bundle of neurons that connects it. And then if the cerebral cortex is the outside part of our brain that you could actually touch, on the inside, right, smack dab in the middle is our subcortical brain regions. So sub, like submarine, like, but think below, like, you know, sub-zero temperatures, like below, like, so subcortical are below the cortex. So the inside portions of your brain. There are different brain regions. There's a lot more than we are going to talk about today, but some of them are really, really important on like telling you like when to sleep, when to wake up, on when you're feeling sexually attracted, when you feel hungry, when you feel like you should be regulating your breaths, regulating your heart rate, things like that. So damage to the subcortical brain regions is really, really bad. And if you damage the subcortical brain regions, people typically don't live, right? If the part of their brain responsible for being their heart isn't working, then their heart stops, right? Occasionally, sometimes people will have brain damage and they'll say, oh, I'm sorry, this person is brain dead. They're physically alive, right? Their heart is still pumping, but they aren't thinking. They themselves, their personality is gone. What's happening is their cerebral cortex, the outside, all the parts that make us uniquely us, are dead or dying, right? Because of like a stroke or traumatic brain injury, for instance. But the inside, the subcortical brain region still could be working. They could still be causing this person's heart to beat, but they will never regain consciousness. They'll never talk again. They won't remember things again, things like that. So anyway, let me share some of the really important subcortical brain regions. The first one is the hippocampus. So the hippocampus is really small, size of like a little cashew or whatever. And it is responsible for memory. Your memories are stored throughout the brain, right? In your subcortical brain, in your cerebral cortex. Meaning during brain surgery, if you were to stick like an electrode in people's brains, they would smell Christmas cookies. They would hear grandma's voice. They would remember what shoe size they wear, like all sorts of random stuff, right? Because your memories are surrounded throughout your entire brain. The hippocampus is a really small portion, about the size of a cashew, in your subcortical brain region, so in the very center. And when it fires, it's saying, we need to remember this. So when someone is, what's it called? In in class, and the professor says, oh, this is going to be on the, this for sure is going to be on the next exam. Your hippocampus is probably going to start going nuts saying, hey, we need to remember this. If you are in Iraq and Afghanistan, you're a soldier, and all of a sudden bombs are blowing up on the side of the road. Your hippocampus is going nuts saying that you need to remember exactly where you were because this area isn't safe. So the next time you come back here, I want you to remember like where the bombs were, where the shots were fired. I want you to remember this because it's really important that you do for your survival. There was a Navy soldier, Navy sailor that went by the initials HM, and he was having bad epileptic seizures, so that they were going to do brain surgery and sever his corpus callosum to prevent, the grand mal seizures. And when they severed the corpus callosum, they accidentally scraped the hippocampus. And what happened is it hurt his memory, right? What happened is when patient HM woke up, he could remember his name, he could remember how to tie his shoes, all of that sort of stuff. He just couldn't form new memories. So what happened is when new doctors would come in and say, hi, I'm Dr. Smith. A few minutes went by and he'd say, oh, like, who are you again? I've never met you before in my life. And Dr. Smith's like, no, like we've been talking for the last 10 minutes. I'm Dr. Smith. Oh, okay. Eventually patient HM couldn't form any new memories. He couldn't recognize himself in the mirror because when he was a sailor, he was 30 years old when he had this surgery. So his memory of himself is when he was 30 years old. So when he turned 40 and 50, he would look in the mirror and get really confused. Who is this old man looking back at me, right? He didn't form any of those new memories of what he was like in his 40s and his 50s. He still thinks of himself as 30 years old. So anyway, the reason why I'm telling these stories is just to really help you guys remember hippocampus is related to forming memories. Other parts of your subcortical brain regions are the amygdala. The amygdala, I want you to think fear, right? When you're watching a horror movie, your amygdala is going nuts. When you're going through like a scary neighborhood or like one of like not scary farms, for instance, and people are jumping out at you left and right, your amygdala is saying like threat, freak out, there's scary people, there's clowns, people are jumping out at you, like you are in a dangerous situation here. I mentioned earlier about the soldier in Iraq and Afghanistan, and the hippocampus was going nuts saying, hey, remember where the bombs were going off? His hippocampus was firing, but also so was his amygdala. The amygdala saying like, freak out, like be scared, be threatened. This is a life-threatening situation. So when the amygdala is going nuts saying, freak out, this is a threatening situation, and your hippocampus is going nuts saying like, remember this, remember this, remember this, what can happen when both of those are going nuts at the same time is the person can develop PTSD, post-traumatic stress disorder. So years later, when they hear fireworks that sound kind of similar to that bomb going off on the side of the road, all of the memories, all of the feelings come flooding back as well. What else do I want to say about the amygdala? The amygdala is active in people that have kind of unconscious racism, right? Sort of implicit or automatic racism. When they feel kind of threatened by people just based on the color of their skin, their amygdala is kind of going nuts. And what's interesting is when you look at the degree of amygdala activation, you can start predicting people's levels of prejudice, how uncomfortable or threatened or fearful they feel in certain situations, which then predicts their behavior. If you were to have a tumor growing on your amygdala, like this guy, Charles Whitman, came back from war, went to the University of Texas as a student, didn't realize he had a tumor growing on his amygdala all of a sudden, just randomly, when that tumor would start to grow, like bigger and bigger and bigger, start pressing on the amygdala, causing it to fire, middle class all of a sudden starts feeling threatened, starts feeling like fearful, anxiety, like bad's going to happen, something, this is a bad situation, I'm going to be under attack. But he was in class. There was no threats whatsoever, but he just felt that there were. Charles Whitman, tumor on his amygdala, caused him to feel that sort of like threat, anxiety, and fear, that he eventually brought a sniper rifle up to the tower in the University of Texas's campus and ended up killing 17 of his classmates. And it wasn't until the autopsy afterwards when they're like, why did he engage in the school shooting? Well, it was the amygdala. He was feeling fear because of this tumor. What else? The brain stem, so kind of connected. Let me show you a picture earlier. Kind of brain stem, things connected to your spinal cord are incredibly important for basic life functions, like waking up in the morning. That cerebellum, that sort of cauliflower thing on the back of people's forehead. Let me show you a picture. Back here, balance. Knowing where like your body is in relation to each other. Are we facing up? Are we laying down? Do I feel dizzy? Kind of things like that. Your cerebellum is involved with things like that. And the way that psychologists know so much about the brain is through advanced measures, right? Finding ways of measuring brain activation. There's a few different like machines, technology out there. The first one is the EEG, electroencephalograph for short. You might have done this if you were on like a sleep study, for instance. And basically what these little sensors are doing are basically measuring like the electrical impulses from all of these neurons around your brain. Because remember, when a neuron fires, it's both electrical and chemical process. So these EEGs are really sensitive to like what parts of the brain like are firing. And because they fire in an all or none response, you get these sort of like parts of your brain are firing, they're not firing, they're firing, they're not firing. And you get this sort of like EEG activity, which is really cool for like sleep studies, for instance. And based on someone's EEG report, you can tell like how, what level of sleep they're in, how deep their sleep is, things like that. But if we want to know more than just, is their brain like, working a lot, you might use more advanced measures of visualizing the brain, like a CT scan, like a CAT scan, right? Computer assisted tomography. Your CAT scan is really basic just for kind of structures. Is there like a tumor growing there? Is part of their brain missing, for instance, your CAT scans? Is our broken bone, damaging the brain, things like that. More advanced would be the MRI, so the magnetic resonance imaging. And basically this is kind of like taking different kind of like picture slices of the brain. And now you can really start seeing all the sulci and gyri. Your MRIs, more expensive, and they're just showing you more details of what the brain looks like. Your CAT scans are really basic. If you want to look at actual function, PET scans are great, positron emission tomography. PET scans over here are just showing like what parts of the brain are more active than others. And then if you lay like your PET scan on top of an MRI, you can also look at the structure from the MRI, but also add in the function and start seeing like what sort of red or kind of hot spots are being used, which is really cool, because then what you can do is put someone in an fMRI machine and show them a picture of somebody that they know, and you start seeing their occipital lobe light up. Or you play them a song that they really like, you see their temporal lobe light up, things like that, which is pretty cool. So that's it. That's it for chapter 2, where we learned all about the brain, lobes, and neurons.